Welcome back to Matrix Uploaded. We're looking at organic molecules, in particular at nucleic acids. Before the break, we said that there are two different nucleic acids that you can find in your body, DNA and RNA. Now we're going to look now at the structure of DNA. We said that DNA was made up of different nucleotides. Those were the packages made up of a phosphate, sugar, and a nitrogenous base. So let's have a look at the picture and see how it is we can put together those four different possible nucleotides to make up DNA. Well, DNA is a double-stranded molecule. That means that we have a chain on either side, very much like the sides of a ladder. And those chains up the side are made up of alternating sugar, phosphate, sugar, phosphate, sugar, phosphate. And just like the ladder has rungs going across it, or steps going across the ladder, so the nitrogenous bases from the two sides of the molecule stretch across and join in the middle. Let's have a look here. We've got sugar, phosphate, sugar, phosphate, up what we call the backbone, and across the rungs or the steps of our ladder, we've got the nitrogenous bases. If we start at the bottom, we can see that our first nucleotide is a phosphate sugar guanine, and that is bonded to a cytosine, which is bonded to the sugar and the phosphate. Now you'll notice that when we bond guanine to cytosine, there are three little dotted lines between them, whereas when we bond thymine to adenine, there are only two little dotted lines. What do those dotted lines represent? Those dotted lines are hydrogen bonds. They're fairly weak hydrogen bonds, but they hold the two sides of the molecule together. Another thing that you should notice is that the cytosine is always bonded to guanine, never to thymine, never to adenine. Likewise, thymine and adenine are always bonded together. No variation allowed. These two rungs of the ladder that are hooked together with little hydrogen bonds and making this double-chained molecule is then twisted on itself, spun around and twisted into what we call a double helix. And that is the basic structure of DNA. It's a large molecule. It has thousands and thousands of these steps or rungs. Complementary base pairing is what we call it when we put guanine and cytosine together. This DNA molecule is an enormous molecule. Not only is it twisted, it is then folded back on itself, and it's almost like it's paper clipped together. It's held together by large protein molecules called histones. Now, this word complementary, please notice how it's spelt. We say that guanine and cytosine are complementary base pairs. They will always bond together. It's not complementary with an I, which is what I would say to you if I said your hair looks very nice today. That's a compliment. This is complementary, which means they complement each other or they go together. And always guanine and cytosine, always adenine and thymine. Now, from what you already know about RNA and DNA, can you predict what a strand of RNA would look like? Well, I think you could already be able to tell me some things. Is that RNA, we said, only has one strand, so you won't find any complementary base pairing. You'll simply have the sugar phosphate backbone with the individual nitrogenous bases sticking out from it. No complementary base pairing. RNA is also much shorter, so it doesn't twist into that helical shape. And also very important, there's no thymine in RNA, thymine is replaced by uracil. The functions of DNA and RNA are very closely associated with each other. We say that DNA carries the code in the nucleus for making proteins. Now, some of the proteins in your body are what we call structural proteins. Your hair, your nails, your bones and skin, and all of those structural bodies within you are made up largely of protein. DNA codes for the making of those structural proteins. But there's other proteins as well. All your enzymes that control different reactions in your body are also made of protein. And so DNA controls the way each of those proteins is also made. And the RNA is going to copy the code from the DNA. It's going to decode the code, and together with ribosomes, it's going to make the protein. And if we have a look at this little diagram, we can see that 1 represents the DNA, 2 represents the RNA, which copies the DNA. The RNA can then leave the nucleus, which is shown by 3. It moves out into the cytoplasm at 4, and together with a ribosome, it makes the proteins, which is shown at 6, which go out into the cytoplasm to do whatever their job is supposed to be in the cell. We'll come back to this picture a number of times when we look at how proteins are made in depth, so don't worry about it. We will cover this once again. What is important from today is that you understand the structure of DNA and the structure of RNA. I look forward to meeting with you again. Thank you for your time. Enjoy going over the work and learning all about DNA and RNA.